40 people are already present. I will start with the webinar. Good afternoon, everyone. Welcome to the 5th webinar on grassland restoration organized by the EXPBIO-Project. I'm Katrien van den Bussen from the Vlaamse Landmaatschappij, where I work in the Department of Agro-Environmental Schemes. The EXPBIO-Project is funded by the EU Interreg North Sea Programme and is a collaboration of 8 partners across Sweden, Denmark, Germany, the Netherlands and Belgium. We are municipalities, universities, botanical gardens, public research institutes and governmental agencies. The EXPBIO-Project is focusing on restoration of grassland biodiversity and bringing back natural flower-rich habitats in the North Sea region. By combining expertise from different countries, we are exploring practical management practices and creating tools to boost the local plant communities. This spring, we kicked off a series of webinars on seed mixtures and seed cultivation. For those who haven't seen them yet, the recordings are still available at the EXPBIO Interreg website. Now we continue this journey with 6 inspiring speakers sharing new insights and proven strategies for the restoration and rediversification of grasslands across Northwest Europe. In the previous 4 webinars, we already saw a whole range of management practices for obtaining species-rich grasslands. And today, this continues with the lecture of Franziska Breit, who is here today to explain how Luxembourg is tackling the restoration of species-rich grasslands. Franziska works as a scientist at the Natuurschutzsyndicaat SICONA in Luxembourg and is responsible for implementing grassland restoration. SICONA has been carrying out grassland restoration projects within his member municipalities for more than 25 years. Over these 25 years, they have gained a wealth of experience by exploring new management techniques from hay transfer to directly harvested seed mixtures to the sowing of cultivated native seeds mixtures. This is combined with long-term and systematic monitoring of the measures for success evaluation. This webinar will last approximately 1 hour with 30 to 40 minutes for the presentation, and afterwards we will have time for discussing. Just some technical matters. I would like to introduce you Gunilla Hell-Bellman, who is responsible for the technical aspects. She will ensure that everything runs smoothly. We also ask you to mute your microphone and use the chat function for communication and questions. With that, I give the floor to you, Franziska.
Okay, thank you very much, and hello to everybody from my side. And also thank you for the introduction and the opportunity to share our experiences in restoring species-rich grassland with all of you. So I will start screen sharing, hopefully. Yeah, I hope you can see my screen. No? Yes, maybe?
Yes.
Okay, great.
Yes.
Okay, I wasn't sure. So, yeah, let's first start with some information about the Nature Conservation Syndicate SICONA. The SICONA was founded in 1989 and is located in the southwest of Luxembourg. At the moment, we have 33 municipalities that are members of the syndicate, and right now we are more than 19 colleagues here at SICONA. Our work is financed by the municipalities that are members, as well as by the state. What's very unique about SICONA is that with our 4 departments, we can not only plan or monitor our projects, we can make every step from planning to realization with our own machinery to monitoring and even raising public awareness by our own. So it's all working together. As we do a lot of nature conservation terms within our member municipalities, we have a lot of working fields like the protection of species, contractual nature conservation, or the creation of standing water bodies. But like the introduction already said, today we will focus on grassland restoration. To set the frame, let's first take a quick look on the current conservation state of grassland habitats within Luxembourg. As for many other European countries, we see that there is a quantitative and qualitative decline in grassland habitats, as well as the remaining ones show a very bad conservation status most of the time. So we can definitely say that there's an urgent need for action. To match this need, there are priorities set within the National Nature Conservation Plan, and the National Strategy for Species-Rich Grassland. The first priority is, of course, the conservation of the remaining biotopes. Second, there's the priority set to improve the remaining habitats as well as to restore habitats. Therefore, the third National Nature Conservation Plan sets up specific targets for optimizing the conservation status, for restoration, and for the creation of new habitats till 2030. When we take a closer look to the lowland hay meadows, this means, for example, that 1,653 hectares of the remaining 2,900 hectares of lowland hay meadows within Luxembourg shall be improved, and that even 2,417 hectares shall be restored. The National Strategy for Species-Rich Grasslands sets up 10 goals to ensure long-term conservation of all existing species-rich grasslands, through extensive land use, as well as through the restoration of species-rich grasslands. So that was just a shortcut to the legal frame. But now, before coming to the real work behind the restoration itself, we can think about some probable problems that might come along with such measures, like the financing, the social acceptance of such measures, the problem with finding the right timing or the right method to fulfill what is your plan to restore. Then there might be problems in monitoring, including the time frame that you need to monitor and the financing of this monitoring, as well as a lack of comparability or a lack of consultation with landowners and managers. I just want you to keep those problems in mind during the presentation. We'll come to it back later. But at SICONA, we use 4 different chains to restore grassland. First, we have the transfer of fresh hay, which we are doing since 2000. Second, we have the storing of directly harvested seed mixtures, which are usually harvested with e-beetle, which is a seed harvester. We're doing this since 2017. Then third, we have the storing of locally produced certified seed mixtures. And fourth, the translocation of rare species. Mostly we perform such measures to restore lowland hay meadows, calcium meadows, Molinia meadows, and other grassland habitats. Independent of which of these chains we're choosing, everything depends on an early planning before starting the practical part and the realization of the restoration. You first have to ensure the financing and apply for all needed authorizations at least 1 to 2 years ahead. Because it always takes time to get the authorization, you need to ensure the financing, because otherwise it's just a waste of money and time in the end. Second, you have to select a suitable recipient site. There you should check before starting anything if the nutrient content is okay, because we're restoring habitats that are specified on nutrient poor places. So if our nutrient content is too high, it's not suitable to perform your restoration right away. You have to do other steps to prepare it, or you have to choose another recipient site. Second, you have to check the soil type, if it's matching the target habitat that you're trying to restore. Third, you have to make arrangement with landowners and farmers. You have to check if the access is guaranteed for the machines that you need to restore the place. You have to ensure the extensive cultivation after restoration, as it has a huge impact on the development of the restoration, because it ensures that it's a long-term success in the end. Then you have also to choose which method you're using. If you're going for the transfer of fresh hay or decide to use seed material. When you're going to the transfer of fresh hay or directly harvested seed material, you also have to find a donor site which matches your needs. Therefore, a donor site register is also very advisable. I will come to this later on. And you should keep in mind that you're taking something from a farmer's site. So you should keep in mind that you might have to provide compensation payments for the donor site or for the use of the donor site. And last one, you should definitely ensure the monitoring and the follow-up care of your restoration, because the process is not finished just because you brought some species-rich diaspores to the recipient site. It's not done yet. Okay, but let's first start with the first steps. We have checked that we have authorizations, that financing is ensured, and we have an appropriate site to do our restoration. So the recipient site is set. Then our first question is, will we do the restoration in stripes or full surface? On established grassland, I would recommend you to make stripes because there's no need to plug up the whole surface. It's enough to go for the stripes. Normally, it's enough to approximately make the stripes on 1/3 of the whole site, and the rest stays as the current sward. Here you can go with the transfer of fresh hay or with sowing seeds. Both is possible. When we do have former arable lands or former scrubbed areas where you have a full surface that's open soil, then I recommend you to go to fresh hay transfer because of the longer streams being a good barrier for erosion, and so it's more suitable for subsiderations. Independent of going for stripes or full surface, there has to be a preparation of the soil of the recipient site in the beginning. So normally we are doing 1 to mostly 2 passages, depending on weather and soil condition. The last passage should be done 2 weeks before restoration. And the one thing that's really important about it is not only that the roots that were on this place are destroyed and dried out, and so there's no problem with reoccurring grasses in that make it harder for the sown material to grow up, but that there's no plugging necessary to open up the soil. It's only superficial tillage that's necessary because you only want to destroy the highest 5 to 6 centimeters. You don't have to plug it. We are normally using our so-called Dutzi to do so. It's a combined machine consisting of a tine rotor and a compact roller. And like I said, it only destroys the top 5 centimeters of the surface. When our recipient site and the soil of the recipient site is prepared, we should think about what material we are using. So when we're looking for a donor site, we have to keep in mind that the target biotope that's present on the donor site is the one that we want to recreate on the recipient site, as well as that the soil type of the donor site and the recipient site is more or less equal to another. Ideally, there is a geographical proximity between the both sites to ensure that these points. Then going for the usage of donor sites, it's also a very good measure to use a donor site register, either to prevent from depletion or degradation of the habitat due to an overusage as donor site, so that you use it too often, too many years in consequence, or in case there are several actors performing restorations within the same area, to prevent double booking of such donor sites, to also prevent from conflicts that may come from this. When we have selected our donor site, we're going for the harvesting. Like all of you might know, we only harvest when the target species are mature. That's easy to go. So when we do the fresh hay transfer, we normally harvest with a tractor with front mower and self-loading loader wagon. And if we go for the directly harvested seed mixtures, we brush out the seeds with an harvester. And yeah, I'll show you that later on. But no matter if you're going for fresh hay transfer or the directly harvested seed mixtures, it's always necessary and very important to think of removing unwanted herbs such as Jacobea vulgaris from the donor sites beforehand, because you don't want to transfer it to the recipient site. So you can just go there, check out, get rid of the flower heads, and then you're fine. But you always have to remove them before harvesting, because otherwise landowners won't like you that much anymore. So think of that. Then taking a closer look to the transfer of fresh hay, the mowing should be done in the morning when it's still moist. This is due to the fact that the seeds remain longer to the stems and don't fall off that easily if they are still moist, because they're sticking a bit better to the stems. But on the other hand, to prevent the hay from rotting, because moist hay rots very easily, the mowing and the transferring to the recipient site should be performed on the same day. To do so, we normally use a loader wagon with dosing roller. Here you have to keep in mind that you have to adjust the driving speed, because otherwise you don't get the uneven spread of the mown material. And that's also an important point. You don't want the mown material to be too thick, neither too thin on your prepared stripes or full surface. Approximately 3 to 4 centimeters are recommended as a layer thickness. Sometimes, as you can see in smaller pictures, it's also necessary to just pat the mown material a bit by hand, because it's not that optimal with the dosing roller sometimes. And in the last step, you should definitely roll your stripes or full surface where you spread your mown material on with, for example, a Cambridge roller or any other roller, to guarantee that the seeds have optimal contact to the soil and can germinate. When we're going for the directly harvested seed mixtures, or e-beetle that we're doing the harvest with it. Here's also a short video that shows you how it works. When we harvested the material, we dry it and stack it till we need it. What's the point for the fresh hay transfer is that we can only do it when the target species are mature, so we're stuck to a short period of time in summer. With the directly harvested seed mixtures, which we tried and have in our stock, we can also perform restorations in spring or autumn. Thereby, we saw the material by hand, because there are some pieces within the mixtures that are still too big to go through a normal sewing machine, so it gets clogged up. So it's not that useful to use it. So we're doing it by hand most of the time. There's also the possibility to use a dung spreader or something like that. That works as well, but we do have the best experiences with sewing by hand. To make the sewing a bit easier, it's also possible to add sand or my spindle meal to the seeds. The sewing density should be around about 10 grams per square meter. And like with the transfer of fresh hay beforehand, it's also here it's important to do a rolling with the Cambridge roller after you sow the seeds, to guarantee the contact to the soil, and also to prevent from wind erosion or something like that. It's always very important to roll. For third pillar is the sowing of locally produced certified seed mixtures. including the complex production of the certified seed mixtures of autochthonous origin. Here's the steps of propagations which are needed to get to these mixtures. Due to the certification, I will come to this later, all production steps are controlled according to strict criteria. First step is the collection of seeds in wild populations according to the M.Sc standards. Then there's the growing of young seedlings in nurseries certified according to Wildpflanzensaison Lützeburg. Then we have the planting and cultivation in fields as monocultures of those plants we just wanted to harvest later on. Then we come to the weed control. That's a lot of work that's mostly done by hand, so it's a very tough step within the whole process. Then we do have the harvesting, which is partly done with specialized machinery. Then we come to the cleaning. This is first done by the participating farmers and done by a company called Riga Hoffmann in Germany, which is specified in cleaning up seeds and also to produce seed mixtures, and that's what they do for us. They make our final mixtures according to specifications given by the SICONA, and that all those steps lead us to the finished mixtures that we have developed yet. So we have several mixtures for the open land right now. Like I already said, there's a certification that leads us to every step being controlled. So the collection, the seedling propagation, the growing of perennials, the production, the cultivation, and the distribution, everything is controlled within this certification process. Therefore, 69 quality criteria are set up for seed production and 38 for cultivation of perennials. The criteria is examined by external institutes and the examination reports supervised by an independent commission. All this certification process is based on the German VWW certificate. Well, that was a short excursus, so let's come back to our real subject. To sum it up, for the practical part at least, we always have the following steps. First, we have the soil preparation. When going for fresh hay transfer, then there's the mowing of the donor site and the transfer of the mown material to the recipient site. And then, as last step, the rolling. When we are going for sowing, independently of directly harvested seed mixtures or locally produced certified seed mixtures, we do have the same soil preparation as for the fresh hay transfer. In case for the directly harvested mixtures, we have the harvesting and the sowing. In case of the locally produced certified seed mixtures, we only have the sowing, and then again the rolling. The three methods have different disadvantages and advantages. Like, for example, the time till the sward will be closed again. When you look down the screen, you see that for fresh hay transfer, we found out that often after around about 3 months, the sward is closed again. When comparing with directly harvested seed mixtures, here it takes mostly 1 year, more or less, till the sward is closed. In contrast, there is a huge advantage for directly harvested seed mixtures as well as for all lax mixtures, and that's the independency between the collection of diaspores and the realization of the restoration itself. Because when you go for the fresh hay transfer, you have to perform the restoration at the same day as the harvesting. When you're going for seeds, you can harvest the seeds even 1 year before you will start your actual restoration. So there are, like I said, disadvantages and advantages for every method, and you have to take a look to your specific situation and choose what suits you the most. What all restorations have in common is that there is the need of a follow-up care. Approximately 2 months after fresh hay transfer or seeding, you should go and take a look at the sites you restored. You should take a look if the target species already occurred, if any problem species for example, room exclusibles occurred, and if there's the need to maintaining cutting or to do pruning to ensure the right development of your restoration. Another point that's very important is the one I mentioned already before. That's the appropriate aftercare and extensive use after restoration. One very good tool for us to ensure an appropriate and extensive use of the restored sites is contractual nature conservation. There we have within Luxembourg different programs which offer models for context regarding extensive mowing and grazing. So you normally can find a good way to get along with the farmers to use the site extensively and to maintain what you've just done. If there's no such program in your case, I would definitely advise you to set up any contractual arrangement before implementation, because that's the only guarantee for you that your work will be successful in the end, because otherwise you invested a lot of time, energy, and money, and there's no secure, nothing that makes sure that it's destroyed afterwards. Yeah, I can't say it different. Well, here's some impressions of how restoration can develop when all the steps that I just told you about have been done correctly. Here we see an example of a fresh hay transfer in stripes. The first picture was taken directly after transfer in 2017, and the second 1 year later in 2018. Here you can clearly see the stripes, especially because of the Acanthemum vulgare, who's shining a bit through. But when you take a look at the same site 5 years after restoration, here in 2022, the stripes are no more visible. The species spread out from the stripes into the surrounding area. And if you take a closer look to the picture on the left side, you also see that many more species have emerged, and not only Acanthemum vulgare like it just looked 1 year after translocation. Here another example for a full surface fresh hay transfer. First, the site has been cleaned of scrub and the surface was prepared. Then there was the hay transfer for the full surface. And 2 years later, you can see a beautiful grassland that can be used as agricultural land, and which developed to be very species rich. Sometimes it's also advisable to use a combination of methods, like in this case, where a restoration of species rich grassland and the installation of a new standing water body was planned, like you can see on the first picture when it's about to be done. So the area used to be covered with spruce forest, and after felling the trees and removing the roads, the first part of the site, here on the picture the back part of the site, was restored by transferring fresh hay. But due to the pond wasn't ready at the time, the front part of the picture was sown with directly harvested seed mixtures later in the same year. Therefore, you see the difference in the development in the first year. The back part over there shows a closed sward, whereas in the front you see a lot of open soil remaining. But when we look at the same site 3 years after restoration, you can see that there's no difference between the two parts. It's one even grassland that can be used. And when we take a look at the fourth picture on the left side on the bottom, you see that it also turned out to be a very species-rich grassland. So the fourth chain for restoring species-rich grassland that we use is the translocation of rare species. I will not talk about this too much because Sandrine just told us a lot about it in the last talk the last week. So I'm just gonna give you some insights. SICONA performs in situ translocations till 2013. Since then, we have planted more than 26,000 seedlings of more than 35 species on more than 100 sites. That's a lot of stuff. Countrywide, there were 558, German misspelling, translocation actions, round about 99 taxes performed within Luxembourg. In general, translocations can function as complementary measures in restorations or as an individual measure in existing biotopes than as a botanical species conservation measure. What you always have to keep in mind when thinking about translocations of rare species is that every species shows different rates in survival and establishment, so every species is specific. You can't just say, oh yeah, I will do it like I did it for Campanula glomerata and it will work out for any other species. You have to keep in mind that it's very species specific. There are a few steps to go through when doing translocations, and they're very similar to the steps I already showed you for the production of the seed mixtures. First, we have the collection of seeds in the wild according to SICONA standards. Then we have the growing of the seeds in nurseries certified according to Pflanzenamt Luxembourg. Then we have the planting of the seedlings. And then there's the difference. We have the measuring with the high precision GPS that is necessary for the following monitoring that is made the first, second, fifth, and 10th year after planting the species to see the survival rates of plantings. Like I already said, for further details to the translocation of rare species, I highly recommend you to take a look at Sandrine's talk if you weren't able to watch it last week. To sum up the practical part, first, you have to set clear objectives and have a very early planning to make sure that everything will work out fine. Then you should have your financing and your authorizations in place before starting anything. Then you have to select a suitable recipient as well as a suitable donor site. You have to make the agreements with the landowners and the farmers. You have to decide if you're going to make a fresh aid transfer or use seeds, or if you're using a combination because it suits your situation best. And you have to ensure that there's a follow-up care to guarantee that there's the opportunity for your restoration to work out well. So what I really recommend is to cooperate closely with the landowners and farmers, because every action depends on their opinion concerning such measures, because it's their land, so we definitely depend on their cooperation. And second, I know I told it often already, but the follow-up care and the extensive land use after restoration are key to the success of the restoration. So always keep that in mind. So now that we've finished the realization part of our restoration, we come to the monitoring part. When the restoration is realized, that's the next step to go to. And this step is as important as the realization itself to document the development of the restoration. Therefore, we at SICONA use a standardized monitoring concept. That means for the recipient and the donor sites, we are making a semi-quantitative estimation of the entire site concerning the species that occur on the site. This is first done before the restoration and then every 6 years. Additionally, on the recipient side, we do install permanent plots from 2 to 8 meters in the restored as well as in the non-restored sub-areas. That's what you can see on the right hand in the picture. The orange ones are the plots in the non-restored sub-areas, and the green ones, those in the restored, in the restorated areas. That's to be able to compare the development of the stripes as well as of the whole site. The monitoring is first done in the year of the restoration and done every 3 years. In difference to the species lists that we make for the recipient and donor sites every 6 years, the permanent plots are there. A cover estimation is made with the extended Brown-Blanquet scale according to Vermont. In 2024, the first evaluation of the results of these monitorings were published. There, the following questions were addressed. Which target species of lowland hay meadows have been able to establish themselves on the recipient sites, and what are their transfer rates in relation to previous land use? How have the number and the cover of the target species and the herb-grass ratio developed in the restored areas? Have the restored areas adapted to the donor areas in terms of their species composition? Have the target species also spread to non-restored areas? And last question, has the habitat typical species composition of lowland hay meadows been achieved in the recipient areas? Therefore, 202 species lists from 43 restorations were analyzed. The data was collected from 2012 to 2022. The parameters used were the number of flowering plants, the number and coverage of lowland hay meadow species, therefore 63 specific species were defined, the number of rare and endangered species, here 10 species were defined, and the average unweighted Allenberg indicator values for moisture, reaction, and nutrients. I will just show you some results of this study due to the limited time, but for further information, I definitely advise you to take a look at Biro et al. 2024, which I will come back to later in further information part. On this graphic, you can see that the comparison of number of target species before and after restoration separated by previous land use. As you can see, there is always a significant difference between the number of the species beforehand and after restoration, independent of the previous use. But you also see that there's at least former grassland not that good performing as the donor sites yet. So there are not that many target species on former grassland than on the donor sites yet. In contrast, former grassland and spruce forests show no more significant difference in the number of target species when compared to the donor sites after 5 to 10 years after restoration. What I missed to say beforehand is that this study is based on results that were from sites where we performed a transfer of fresh hay. So it's not no sown materials, only for fresh hay transfer. On this graphic, you can also see that the classification of the sites in terms of their status as lowland hay meadow increased due to the restoration. For all the sites, you can see an improvement concerning their ranking mostly to level A. So that means that there were more than 50 target species occurring into these sites after restoration. Especially for former farmland and spruce forests, which started at a point of not even being any grassland, the results are very impressive, as they all reached at least level B, but mostly level A after restoration. When we take a closer look on the transfer rates of the target species, we see that on average, 45% of the target species from the donor sites were transferred to the recipient site when the recipient site was an established grassland. When looking on the transfer rate from donor areas to former arable land, there was on average a transfer of 76% of the target species. Taking a even closer look and going to the individual target species themselves, we see that there are some species which are successfully introduced to nearly every restoration site, that are the ones on the left hand that were transferred from 100 to 61% of all restorations. And on the right hand, you see that there are also species which were nearly never transferred, like Campanula glomerata, which occurred in only less than 1% of the recipient sites after restoration. This brings us to the conclusion that rare species are less easily transferred and must be introduced in a targeted approach. So that's where we're coming back to the translocation of species and their need to complete the species inventory of biotopes. To sum up the results for the study, we can say that species richness increased on recipient sites, that the restoration of former arable land and spruce forests is more successful than on grassland sites, that the transferability of target species depends on many factors, for example, the frequency they occur on the donor site or the time when the seeds are mature, and that the transfer rates are on average 45% on grassland sites and 76% on arable land. We also see that rare species are less easily transferred and must be introduced in a targeted manner. And I know I didn't show you the clear results for that, but it's also listed in this paper. So I just wanted to mention it anyways, because it was shown that the restorations older than 7 years are more similar to the donor sites than the younger ones, and that most target species were able to spread out of those stripes into the non-restored areas. We all know that it's a slow process, but it's working. And that's something very important to keep in mind. It's a slow progress, but it's working. So we can say that the restoration of mesophilic grassland due to fresh height transfer was definitely successful, as species compositions achieved the rating A for lowland hay meadows. So it was definitely a success to restore those sites. All this clearly shows us that you should always include monitoring, as it is essential for checking the quality of the measures and also to be able to make adjustments if necessary if you see that anything is working out wrong. We can definitely advise to use a concept that is based on more than 10 years of experience. So we're glad to have these standardized methods. And although you might not use the same methods as we do, it's important to come to the point that we have uniform standards for monitoring success, as it is important for the evaluation and comparability of the success and the restorations themselves. So to come back to the possible problems mentioned at the beginning, there are some lessons learned to avoid them. At least I hope so. But you should always keep in mind is the timely planning of the whole restoration, from the financing and the needed authorizations to the monitoring. That's definitely very, very important to avoid so many problems that can make it way more difficult for you to realize your measures. You should always evaluate the success of the restoration, and this should follow standardized methods to be comparable and just to ensure that it's really working out and that you can what you should always keep in mind with the monitoring as well is that it needs time. Plants aren't that fast to spread around or to even emerge, so it's insufficient to do monitoring for 1 to 2 years. we definitely advise you to do monitoring for up to 10 years. And one thing that's very important to me is that you have to keep in mind that you're depending on the farmers and the landowners. So always cooperate with them closely, and that right from the beginning, not when you are near to realizing your measures. Really talk to them in the beginning, it's way easier. If you want to get further information, even international information on this subject, I want to recommend you the Netzwerk für Naturierung. It's a network with an email distribution list and more than 400 members. The network serves as a synthesis between science and practice and promotes the exchange of experiences in the field of restoration. I also encourage you to take a look at some publications about the subject, which you can find on our website and are also listed here. Yeah, I guess that's all from my part. If there are any questions, just go for it. Otherwise, thank you for your attention.
Thank you very much, Franziska, for your inspiring speech. So we can go to the questions. There are some questions in the chat about the practical implementation of hand sowing. How can you guarantee that the right amount of seeds ends up on the intended area?
We tested it beforehand, so we just checked out more or less how much of seeds is inside those grams. And there are also some papers dealing with this subject. So it's literature based and some self-experience. And we have, if you go for seeds that you buy at the storage, there's normally around about 3 grams per square meter to have sufficient seeds on the place. Due to us having more larger material still inside our mixtures because of the harvesting and steams coming with it, we're going for 10 grams per square meter to have sufficient seeds. I'm not quite sure if I got the question right, but I hope I could help. Otherwise, the concerned person might ask again. I'm sorry.
The next question, why do you only use phosphorus content to choose the most nutrient-poor sites? How about nitrogen?
That was, I didn't mention that, but we do take into account the phosphorus rate as well as the nitrogen rate. Both is important, but more limiting is mostly the phosphorus rate because you can get rid of nitrogen, but it's tough to get rid of phosphorus. So that's the more limiting factor, because if you have too much nitrogen inside in the soil, you can go for intensive mowing, or you can, in case you have some farmland, you can stop fertilizing and still use it as farmland, so you get rid of the nitrogen, but the phosphorus is mostly fixed to the soil. So you have to remove the topsoil, or you just have to handle the high loads of phosphorus.
Do you remove the inoculated hay after a while? If so, when?
We don't remove the hay because it would be, on the one hand, a disturbance to the occurring seedlings, and there's no need to remove it because it's watching anyway, and as long as it's present on above a seeds, it keeps the moisture a bit longer. It's protecting a little seedling, so thus there's no need to remove it.
And how much hay do you put on the receptor sites?
Normally we go for an equal distribution. So if we have a donor site from about 1 hectare, we can restore 1 hectare with this material.
And then another question, could patches be used rather than stripes? Might look more natural in the landscape, but less handy for using machinery, I guess, is the question.
I would say the question's already answered, but I would definitely go with that, right? Sure, patches are possible. It's more work for us, so it's not that good to handle. So we're going with the stripes, but sure, if you have the capacity to do patches, go for it.
So that's the last question. There is coming another question. I think the following usage of the restored areas is essential. Do you have experience with special mowing techniques? For instance, 10 centimeter high, classical mowing, or with balken?
Okay, I guess that's a German question. Yeah, so we don't have specialized mowing techniques due to our intention is that the land, that the farmers themselves can make can still use their sites as they were used to. So there are restrictions within the contractual nature conservation with the dates that they are allowed to mow. So we start mowing at the 15th of June, but there's no restriction of the height that they are mowing. We recommend to do a height of 10 or more centimeters, but there's no possibility to tell them you have to. So when we are doing the mowing ourselves, we go for a height of 10 centimeters, and we usually use the balken nail. I don't know the English word either, I'm sorry. Yeah, that's what I can say to that.
And do you have grasslands where you implemented extensive grazing after restoration?
We do, because the contractual nature conservation is the farmers can choose freely which contract they want. So if they want to go to do grazing one, that's fine for us as well. But we don't see that, we don't like it that much when they go for grazing, because Luxembourg is a tiny country, so our sites are very tiny too. And if you restore this problem with in the first year, there shouldn't be any grazing on the stripes or on the surface, because it's not working with the seedlings to grow if they're just trampled by the animals. So we would have to fence them out, and that means there's nearly no remaining space for cows or any animals to graze on. So we do have some grazing on our restored sites, but we don't recommend it in our cases.
Another interesting question. You showed the establishment rates of the different grassland species. Do you also have a dispersion rate out of the stripes into the existing grassland, or any indications which species disperse more easily?
I must admit that it's a study of a colleague of mine, so I'm not quite into every detail, but as far as I know, there are no information about which species disperse more easily or quicker or anything. I guess that's something for future analysis of our data.
I think that it's the last question in the chat. Now there is coming another question. In the UK, we always recommend grazing after mowing to keep the grass short for the spring germination, but it is milder here and the grass keeps growing all winter.
Yeah, I don't think so for Luxembourg, to be honest. And we often have the case that yeah, no, it's not the case for Luxembourg, I guess. We're definitely going for the Marine first, but yeah, we have tiny spaces as well. So, but it's interesting to hear that they are doing different in the UK, definitely.
Okay. So I have a question myself.
Sure.
I think it's impressive what SICONA has achieved in those 25 years. And what do you still see as the main challenges for the near future?
Stop sharing them. The main challenges for the future are to find appropriate recipient sites. As I showed you in the beginning, there are targets set by our third National Nature Conservation Plan to restore a lot of stuff in, when you think of how big the whole country is and how much they want us to restore. And it's not that easy to find appropriate sites, either due to the nutrient load of the sites or to the acceptance of the farmers, because it's always combined with the extensive use of the site, and that's not what every farmer is willing to do. So I think the most problematic part is to find appropriate recipient sites, because the donor site problem is more or less solved with a production of autochthonous seed mixtures. That helps us a lot, because otherwise the donor site acquisition would be a problem as well.
Thank you. There's another question. Do you have a minimum number of years the farmers must maintain their restored grasslands?
Yes and no. When we have consequent contractual nature conservation that follows the restoration, this takes 5 years, and then he has to renew it if he wants to. So the 5 years of extensive use are definitely set, and then he can rethink about it. And if we're doing any other contracts with the land users or land owners, we try to go for 10 years, but yeah, it depends a bit.
Okay, thank you very much, Franziska. Next week, there will be no webinar, but I hope to see you all again in 2 weeks for the final webinar on grassland restoration given by Johannes Koolman from the Technical University of Munich. He will give a presentation on the establishment and effects of flower strips in arable landscapes. He will tell us about his research on the diversity and interaction of plants and insects in contrasting types of flowering strips. If you would like to attend this webinar, please register. All webinars are recorded. The first 3 are already available on the Interreg webpage. After this whole webinar series is accomplished, you will receive an email with a questionnaire on reflections and learnings. We would greatly appreciate if you would fill this in, as it really helps us to make recommendations for future steps in grassland restoration. You can still use the chat for another few minutes if you have any remaining thoughts to share, and we will check it afterwards. So, on behalf of the entire EXPBIO team, I would like to thank you all for participating, and you're welcome to the final webinar. See you then. Bye.
Thank you. Bye.